Hi class. This lecture is going to be all about transcription and translation. So we've already learned about the structure of DNA, and now what we're going to do is kind of go a little bit deeper into the purpose of DNA and all of like the key players that end up making protein. And so the process is also called gene expression. And there's two stages to it. There's transcription and there's translation. And it involves...
Hi! I'm cool!
Zoe. It involves obviously DNA, and if you remember from like September and October, we learned that DNA is located in the nucleus of the cell, and it does not leave the nucleus, if you remember, but it does have pores. And those pores are actually where the information that DNA has gets sent through those pores through another type of nucleic acid called ribonucleic acid, or RNA. Yes?
Sorry to interrupt, but I need a box. Like, know how you put this in a box? That thing?
You have to look in the top drawer.
They're not.
So anyways, what are these like similarities and differences of RNA versus DNA? So one unique thing is that as far as the strands, RNA is a single strand and DNA is a double strand. Another key thing that's different is that RNA actually has the sugar ribose, hence the R in RNA, and then DNA has the sugar deoxyribose, which is the D. So that's another key difference. And then another thing is that RNA has these nitrogen bases: adenine, uracil, guanine, and cytosine. You can notice here that uracil actually is it's a different nitrogen base compared to DNA. So with DNA, remember, adenine pairs with thymine and guanine pairs with cytosine from our DNA song. Adenine and thymine make a lovely pair. Cytosine without guanine would feel very bare. Okay, so those are those nitrogen bases, but when DNA has to be transcribed, it actually gets transcribed into RNA. So wherever like there is an adenine in DNA, you'll notice that here there's no thymine, so you would use a uracil instead of thymine when doing the pairing, and we'll get into greater detail with that in a minute. Okay, so make sure you write all that stuff down. So the first step, again, is called transcription. And this basically happens when DNA that's inside the nucleus gets unzipped, and then we have an enzyme called RNA polymerase that comes over and actually binds nitrogen-based pairs to the DNA template. Okay, and the way that the enzyme helps to do that is with messenger RNA. So there's different kinds of RNA that we're going to have to learn about. So messenger RNA is the one that's actually in the nucleus, and it reads what the DNA strand is saying. So maybe it says cytosine, guanine, adenine. So if it's cytosine, guanine, and adenine, what mRNA will do is it'll transcribe that, and that means that it would bring a G, because C pairs with G. It would bring a C because it's transcribing what this DNA is saying. And then for A, we naturally want to write a T, don't we? But no, we write a U instead, because RNA has uracil instead of thymine. So this transcribed section right here is called a codon, okay? So a codon is a 3-letter, 3 nitrogen base kind of package deal. And that's as a result of transcription. Transcription occurs in the nucleus, okay? Then the next part is called translation. And so what happens is mRNA, which is the RNA that transcribed the DNA, moves through the cytoplasm to a ribosome. And if you remember, our ribosome dance routine thing was this, which meant hamburger, which means protein. So the mRNA travels to the ribosome. And what it does is it attaches to the ribosome, and specifically there's another RNA there called tRNA. And tRNA actually helps to translate this information into a protein. Okay? So when it goes to the ribosome, it actually is going to another kind of the RNA called rRNA. So this next slide actually goes over all the different kinds. So we've already talked about messenger, and that's the one that is in the nucleus. It's made during transcription, and then it actually goes out of the nucleus through those nuclear pores, through the cytoplasm to the ribosome, and specifically when it gets there, it gets greeted by ribosomal RNA. And ribosomal RNA will basically help with translation. And then what happens is there's another RNA friend called tRNA. And tRNA will actually match the correct amino acid that gets translated from that sequence, okay? So when I talk about an amino acid, you guys, what I'm talking about is this table that we're going to be become very used to this table and how to use it. Basically, for translation, you need to come up with an amino acid from the 3-letter sequence. So if we go back to this one up here, we had transcribed this into GCU. Then in order to figure out what GCU makes, so we'll write that here, well actually it was an RNA. GCU was the RNA. So the DNA counterpart of that was CGA. And so then DNA gets transcribed into RNA, and then the RNA gets translated into an amino acid. So how do you figure out what amino acid it is? Well, this side here is all of the first letters. This here is the second letter. And then this is the third letter. So you read the table left, top, right. So if our first letter is G, here it is right here. Our second letter is C, and here's that, and we're actually going to move down and meet the G. And then our third letter is U. Here's our U. What we do is kind of connect it all. So GCU makes the amino acid alanine. I'll just put ALA for short. So that's just one amino acid. But what we know is that proteins are actually made up of many amino acids. So when you put all of those amino acids together, it'll make any type of protein. And we know, like in cellular homeostasis, that proteins are so needed in so many instances, from enzymatic roles to maybe proteins that are like channel proteins or carrier proteins that are in the cell membrane to help maintain homeostasis. There's just so many roles that protein provides in living things. So in order to make that protein, that information is actually found in DNA. So I think that's pretty cool, because usually when people think about DNA, they just think of like phenotypes, like physical traits, curly hair, straight hair. If you've ever seen different types of breeds of chicken or breeds of dog, like everybody thinks about those things and how they look, when in essence DNA plays a much more intricate role when it comes to protein production. And so transcription and translation helps us better understand that process. All right, good to go.